Lung Regenerative Therapies by Katrina Griffith in SportsMed 3, taught by Isaiah Tanasi. First, I will talk about IPF, and I will be talking about the new therapies for IPF. Then, I will talk about cystic fibrosis, and I will be talking about the new therapies for cystic fibrosis. Third, I will talk of recellularization of decellularized lungs, and I will also be talking about the new therapy including this. Also, with cystic fibrosis, there is going to be talk of stem cells. First of all, what is IPF? Idiopathic pulmonary fibrosis, IPF, is the most common type of pulmonary fibrosis that causes scarring of the lungs. The word idiopathic means it has no known causes. So, scarring causes stiffness in the lungs and makes it difficult to breathe. So with people who have idiopathic pulmonary fibrosis, they usually do not know the reason for it, and it makes it very hard to diagnose. New IPF therapies. Researchers have identified the specific signaling proteins that go awry in the fibrotic lung. So, if mice with lung fibrosis inhaled a lab-grown version of a key protein called bone morphogen protein 4, BPM4, it could reverse the behavior of the stem cells, restore normal lung regeneration, and improve lung function. This also presents a new therapeutic paradigm for lung fibrosis, where the goal is not to just stop the scarring, but also stimulate native stem cells toward a regenerative program to restore healthy lung. Now, what is cystic fibrosis and the new therapies? So, cystic fibrosis is a disorder that causes severe damaging to the lungs, digestive system, and other organs in the body. Cystic fibrosis affects the cells that produce mucus, sweat, and digestive juices. These secreted fluids are normally thin and slippery. But people with CF, a defective gene, causes the secretions to become sticky and thick. Instead of acting as lubricants, the secretions plugs up tubes, ducts passageways, and especially in the lungs and pancreas. Also, cystic fibrosis is inherited, so it does not come from outside sources. It's nurture. New cystic fibrosis therapies with stem cell engineering. So, through cystic fibrosis with the therapies, exomes are nanized vessels that are naturally produced by virtually all cells and are involved in cell-to-cell communication. Based on the use of engineered stem cell exomes as a regenerative carrier for inhaled gene therapy. Recent research highlights the potential of stem cell exomes as a transformative regenerative therapy and medicines. The engineered exome platform will be used to develop a first-class regenerative gene therapy for cystic fibrosis. The process is for the exomes to efficiently penetrate mucus and enter into the targeted lung cells, demonstrating widespread and efficient entry into high number of cells that are required for truly successful cystic fibrosis gene therapy. Continued. In the future, it is hoped that this platform will provide useful treatments for treating COPD, lung cancer, and many other lung diseases. This enhanced delivery also has the potential to maximize the regenerative effects mediated by the stem cell exome. And inhaled bioengineered exome therapeutics delivered by a tailored aerosol delivery method based on vibrating mesh nebulizer technology gives hopes for new therapies and help cure of cystic fibrosis. So, recellularization of decellularized lungs. This is a new practice with new research to try to enter recellularized lungs into a donor lung to be transferred back into the patient. So, for Mayo Clinic researchers, they are studying lung decellularization and recellularization techniques to move forward toward a goal of on-demand production of patient-specific transplant-ready lungs. What this means is lung decellularization involves removing all the cells from the donor lung, leaving behind just the architecture that can be recellularized with induced pluripotent stem cells, acquired from the patient's own tissues. Researchers are now working to generate a functioning lung that is suitable for transplantation by first recellularization of a porcine lung. Then, Mayo Clinic investigators are now attempting to recellularize human lungs. Ongoing work involves differentiating iPS cells into lung-specific cells for recellularization. So the new hope is that they can just decellularize the donor lungs and recellularize them with their own lung cells, which means they won't need for any type of medicine in order to not reject the lungs, because they will be seen as their own with their own cells. Thank you. If you have any questions, you can contact me through Sports Medicine 3.